All right, today I'm going to review more DNA technologies, specifically two new ones, genomics and proteomics. Those are two that we haven't talked about before. And these use similar technologies. They're just whole genome, whole proteome, and proteome means all the proteins produced by an organism. And so what we see here is a Neanderthal skull and skeleton and looking at the how much DNA is present there from uh, Neanderthals versus humans. And I'll explain how this works later. It's just an extension of gene sequencing technology. So the last couple times we've been talking about various technologies that are kind of lumped together as DNA, biotechnologies, genetic engineering, things like that. One of the earliest was cloning a whole organism, where we just replace the nucleus of an egg cell with the nucleus of a body cell, and then allow that egg cell to divide, and we implant it in an organism so that we can get a developed embryo that will give us a cloned organism. Uh, that was one of the earliest technologies. Another early technology that's widely used, probably one of the most widely used ones, are recombinant DNA, recombinant cells. Here we use enzymes like restriction enzymes and DNA ligase to cut DNA segments and join them together again. And so we can take DNA from uh, uh, a source organism and put it into a host organism. And that organism, transgenome organism will produce the proteins that were normally produced by the source cell. And so we use this to produce human medications, to move genes into crop plants, all sorts of things like that. It's been very, very effective. Um, Again, one of the early ideas was gene therapy, where we could cure genetic diseases in people by giving them a functioning copy of the DNA that they're missing. And that way their body would produce the materials that they needed. This has been less successful, more difficult to get to operate than other technologies. The problems of getting the genes into the right cell, not in the wrong cell, and getting them active and back in the host has been more difficult than people originally envisioned. Then we have a bunch of DNA technologies that we talked about last time that basically all use DNA polymerase, and we get DNA to be copied in test tubes. The first one of these to be really commercially available was polymerase chain reaction, where we would use heat to separate DNA strands and then the enzyme from a hot springs bacteria that operates at high temperature to copy the DNA. And we cycle between the temperature that DNA strands come apart and then the temperatures that the enzyme needs, and we can copy it with each cycle. And using this, we can produce unlimited copies of any DNA from any source. Very handy, very nice. Okay, then we talked about automated DNA sequencing. Here we use the same enzyme, DNA polymerase, with some nucleotides and then some chain terminators that are linked to fluorescent dyes. And we get a bunch of fragments, each one ending with a different colored fluorescent dye. And by separating these by size and reading the colors with a laser, we can automate the sequencing of a whole genome. Uh, the original Human Genome Project took like 10 years and billions of dollars. Now we can sequence DNA, pretty much a whole genome in a single afternoon for maybe $10,000. Okay, and then I ended up last time talking about DNA profiling, where we can identify individuals by unique segments of DNA, either the short tandem repeats that we can find using probes, or we can cut their DNA up and get a pattern by separating the fragments, and that's what's shown on the bottom here. It's one of the easiest ways to do DNA profiling, and this gives us very specific, very unique patterns for each individual, and so it allows us to identify individuals quite confidently. And it's been widely used, and I talked about how it's used in the justice system, much better than previous technologies that were based on things like blood type, even fingerprinting, which requires a lot of luck. The unfortunate thing here is this has shown us that our legal system was giving us a lot of um, false convictions. And so uh, we've spent the last 20 years kind of really reviewing lots of convictions and overturning them and getting new convictions. Okay, so the new technologies we're going to talk about are genome mapping and gene editing and things like that. Genomics is mapping all the genes on a chromosome, getting a physical map of the chromosome, knowing what genes are there, knowing what genes are involved in various diseases. Um, the Human Genome Project was our first example of this, but we've done lots of improvements there. We have something like 3.1 billion base pairs on 46 chromosomes. Um, and of on these 3 billion pairs, we can identify something like 22,000 protein encoding genes. And most of these genes, we really don't know what they do yet or everything they do. We have a general idea for many of them. Um, some of them we know are involved in specific diseases, and some we know very well. But many of them we don't really know at all. But we may know them sometimes when something goes wrong. Um, so there's a use in these is being able to identify rare genetic diseases and disorders. And so our book talks about several cases where there are unusual cases, and there's no real recourse to treatment other than repeated kind of random tests. But once we've done genomic testing, we can identify unique differences in an individual from previous segments, and that can really help us. Um, this, these whole genome mapping things, these are done with the DNA sequencing machines, and we do them and reconstruct them with computers. With metagenomics, we can actually take DNA from a source like soil or uh, maybe a biofilm that's lining a tube, and we cut up all the DNA and then sequence it from all these individuals. And using computers, we can reconstruct sequences of individual species. And so we can learn what species are present in a multi-species area, um, and kind of have an idea of how they interact. This has been very important when we're talking about things like, um, uh, how organisms process pollutants in the environment, because often one organism only processes it so far, and it'll rely on another organism to take it further. We also see this technique, like right now in archaeology, where they're excavating caves that humans occupied at some point. They can go back and reconstruct the genomes of those people from just taking soil samples at different levels. They can find enough DNA in there to reconstruct genomes. They can identify um, them to species and maybe even to various types and compare them with modern people. So very, very powerful. And now that we have this whole genome sequencing and we can identify specific genes and have a better idea what genes are doing, we actually have tools for editing some of those genes. And so we can sometimes go in and using very specific tools, um, a series of enzymes called CRISPR, which do a site-directed editing of the genome and can install different sequences in those sites. We can go and change this, and we can particularly remove genes, often in early embryos or sometimes later. Um, let's switch now to talk about proteomics. Proteomics are looking at all the proteins that are produced by an organism or of a group of cells in that organism. So we can, now that we've identified all the proteins by looking at the gene sequences that produce those proteins, we can identify what cells are producing what proteins. And this has been very useful in cancer treatments because we can see, looking at tumors, we can identify the proteins that those cells are producing, and we can compare those with normal cells and look at the differences. We can possibly go back and edit those cells to see if we can change them. And we can get an idea of, um, by knowing what proteins are present, we have a pretty good idea of what treatment options we should have. So again, these are new fields growing rapidly. The uses are expanding. Uh, there will be more uses of them as we go. So, um, just to summarize, DNA technologies allow us to sequence DNA from small samples. We can identify DNA from mixed environmental samples. We can identify and model all proteins coded for the DNA, and we can identify which proteins specific organisms or specific cells are using. Really powerful technologies that allow us to look at cells in new ways and organisms in new ways.